For decades, when we've talked about Alzheimer's disease, the conversation has really been dominated by two proteins, right? Amyloid and tau. But what if they're just the smoke, and the real fire is something deeper, a metabolic crisis happening inside the brain? Today, we're going to dive into a major paradigm shift in Alzheimer's research, one that reframes the disease as a case of profound neuronal starvation.
And this quote just sets the stage perfectly. It reframes the entire problem. This idea, which is really gaining a lot of traction, suggests that maybe we've been focusing on the downstream effects, while the root cause is a fundamental failure in how the brain actually produces and distributes its energy. We're moving from a purely protein-centric view to a bioenergetic one.
So to really get a handle on this collapse, you first have to appreciate the brain's incredible energy demand. It is a true metabolic paradox. I mean, think about it. This tiny organ, just 2% of our body weight, is an absolute energy hog, burning through a quarter of our body's total energy. And here's the thing, neurons can't meet this demand all on their own. They absolutely rely on their support cells, the astrocytes, to act as a metabolic lifeline, constantly feeding them the fuel they need to function.
Okay, so here's our roadmap for this explainer. We'll kick things off with this new bioenergetic failure model. Then we'll look at how a healthy brain gets its fuel. After that, we'll introduce a key enzyme, IDO1, and the molecular chain reaction it sets off to starve neurons. And finally, we'll explore the devastating consequences and, importantly, a potential way to restore that metabolic lifeline.
So this new model really flips the script on the sequence of events. Now, this doesn't mean amyloid plaques and tau tangles are irrelevant. Not at all. They are absolutely key pathologies. But what the evidence is starting to suggest is that an underlying metabolic crisis actually comes first. It not only precedes their buildup, but it also makes the brain way more vulnerable to their toxic effects. The energy failure is the first domino to fall.
All right, so to understand what goes wrong, we first need a clear picture of how the brain's energy supply is supposed to work. And the foundation for this is a really beautiful piece of metabolic teamwork called the astrocyte neuron lactate shuttle, or ANLS for short.
Okay, what's so cool about this slide is it breaks down this incredibly elegant system. You see, astrocytes are perfectly positioned right next to blood vessels, so they can just grab glucose as it goes by. Then, through glycolysis, they convert that glucose into lactate. Now, this lactate isn't waste. Think of it more like a high-octane fuel that they then shuttle out to nearby hungry neurons. The neurons take up this lactate, convert it back to pyruvate, and use it to generate the enormous amounts of ATP they need for literally everything they do. It's a perfect division of labor.
So if that's the healthy state, what breaks it? What throws a wrench in the works? Well, a lot of evidence is pointing to one key pathogenic trigger: the overactivation of an ancient immune enzyme called IDO1. This is the villain of our metabolic story.
So what exactly is IDO1? Well, it's the key, rate-limiting enzyme in a pathway that breaks down the amino acid tryptophan. Its main job is to produce a metabolite called kynurenine. And the crucial detail here, as you can see, is where it's found. It's expressed in astrocytes and microglia, the brain's support and immune cells, but not in the neurons themselves. And this is where the classic Alzheimer's pathologies make their dramatic re-entrance. Amyloid beta and tau act like chronic inflammatory signals. The astrocytes sense this danger and go into a kind of defense mode, which involves dramatically ramping up their IDO1 activity. This creates a massive flood of kynurenine inside the astrocyte. And this is where the metabolic hijack begins.
So this brings us to the core mechanism of the whole process. That flood of kynurenine doesn't just sit there. It actively sabotages the astrocyte's ability to produce energy. It literally shuts down glycolysis from the inside out. So, the big question is, right, how does the activation of this enzyme in an astrocyte actually lead to the starvation of its neighbor, the neuron? To figure that out, we have to follow the molecular cascade step by step.
Okay, this slide is brilliant because it lays out the molecular sabotage perfectly. That flood of kynurenine, it activates a receptor called AR. AR then heads to the cell's nucleus, but it can't work alone. It needs a partner, a cofactor called ARNT. Now here's the catch, and this is the whole key. ARNT is a limited resource. And another critical factor, HIF-1 alpha, which is the master switch for turning on glycolysis, also needs ARNT as its partner. So what happens? The activated AR basically outcompetes HIF-1 alpha, hogging all the available ARNT for itself. Without its partner, HIF-1 alpha is powerless. It can't turn on the genes for glycolysis, and the astrocyte's entire lactate production line just grinds to a halt. And the result of this molecular competition is direct, and it is absolutely devastating. That metabolic lifeline, the one connecting the astrocyte to the neuron, is effectively cut.
This comparison just lays it all out, crystal clear. On the left, you've got the healthy state, our functional lactate shuttle, where astrocytes are busy little factories, churning out lactate to feed neurons. But on the right, in the pathological state, IDO1 has hijacked the system. The glycolytic engine is off, lactate production is stopped, and the neuron is left stranded without its primary fuel source.
And look, this state of metabolic starvation is not a subtle thing. Without the ATP it needs from lactate, a neuron can't maintain synaptic plasticity, which is the very basis of learning and memory. This energy crisis translates directly into synaptic failure and then into the cognitive decline that we recognize as the hallmark of Alzheimer's disease.
But, and this is a really important but, the research doesn't end on that bleak note. If this metabolic collapse is a central part of the disease, could it also be a target for a therapy? The findings here are actually incredibly hopeful because they suggest this whole process might just be reversible. So researchers decided to test this hypothesis directly in preclinical models. They came at it from two angles, either by genetically deleting the IDO1 enzyme entirely, or by using a specially designed small molecule drug that could get into the brain and block it. And they tested this in mouse models that had both amyloid and tau pathologies. The results were, well, they were just striking. This chart shows the lactate production in astrocytes. So, in the Alzheimer's model mice, as you'd expect, it's really low. Healthy controls are high, but the crucial bar is the one in the middle. In the AD mice that got the IDO1 inhibitor, lactate production was restored almost completely, right back to healthy levels. It's definitive proof that blocking that hijack mechanism actually works. And here's what's even more profound. This metabolic rescue had real functional consequences. Restoring the astrocytes' ability to make lactate didn't just fix a number on a chart, it actually rescued synaptic plasticity and reversed the memory and learning problems in these animal models. They got better, quite simply because their neurons were finally being fueled properly again.
And so this all leaves us with a really powerful and provocative final thought. We know that neurodegenerative diseases, from Alzheimer's to Parkinson's, they have different protein culprits. But what if many of them ultimately converge on this same common pathway of metabolic failure? If that's true, then targeting that failure, restoring the brain's metabolic flexibility could be a powerful unifying strategy to treat them all. It's a brand new and incredibly promising frontier in our fight against these devastating diseases.